Hi guys, my name is Jasmine Espinoza. I'll be doing my research project on the microorganism called CaNDiDA albicans. A common name for my microorganism is also thrush. On the left side over here, this is a picture of my microorganism, what it would look like under a microscope. Next would be the article that I used for my research project is titled "How Does Temperature Trigger Biofilms Adhesion and Growth in CaNDiDA albicans and Two Non-CaNDiDA albicans Species." The authors of this research article were Deborah Casadegrande Pierre, Laura Corte, Arturo Casadevall, Vincent Robert, Gianluigi Cardinali, and Carlo Toschini. This research took place in the Department of Pharmaceutical Sciences at the University of Perugia Borgo, which is located in Perugia, Italy. This research was taking place on the date of February 19th of 2021. So for the first page of my research project, which was the abstract of this research, basically to give you some more insight of what this presentation is kind of going to be about. So a group of researchers knew that bioformation on basic and aerobic surfaces are finely regulated by genetic factors which contain oxygen, pH, or temperature. Those are some environmental factors that take place when a biofilm is formed. They wanted to kind of know, based on the temperatures of different species, including my species, what the reaction would be, which ones would survive and which ones would die, or which ones would grow, or which ones would not grow, which ones would stay the same. A little bit of the background of my research. So, for starters, biofilm formation are mainly regulated by factors such as oxygen, pH, temperature, just to kind of name a few environmental factors. Um, what is known as for biofilms of Candida pathogenic species is among these species, they can survive in extremely cold and extremely hot temperatures. Also, the resistance to tolerance for a higher and lower temperature can threaten fungi species in the epidemiological level. The study is basically about thermal tolerance and of the biofilms that are formed from the three different species. One of them, again, being mine, my CaNDiDA albicans. They kind of wanted to know what the reaction would be between these different species. So the methods for this were the first one was that 30 non-nosocomial strains were investigated on their abilities in high and low temperatures. And then they were actually isolated and then placed in an incubator at -37 Celsius for 24 hours. So the biofilms were actually tested in micropipettes and then they were inoculated. The researchers gathered the two worst and the two best that were affected by the temperature change, and they kind of researched it a little further to kind of see what exactly is different from the two best and the two worst. And for my graph, so basically, this graph kind of shows each biomass produced by all the strains at each test temperature. So on the x-axis, you see the temperature of each biofilm, which it was placed at, which is here. And then in the y-axis, you can see the amount of time each biofilm was placed in a certain temperature. Throughout this experiment, all species displayed a large variety of growth from different temperatures, which was surprisingly because they were expecting some to do good and some not to do as good. And then overall, so basically, this research paper From what I gathered from my research article was that biofilm biomass produced at 37 Celsius showed that the strains grew in size within actually just 2 days, which was overall the same for all the other three studied species, which the researchers were not expecting to find out when they conducted this research. When the temperature decreased, it looked like it stopped the growing of the biofilms. Interestingly, the growth at 34 Celsius or 31 Celsius was greater for my species for this overall project. And yeah, that was a little insight of my research project. I hope you guys liked it. And yeah, bye.